Lo que se acerca un caballero, su peluña pintaba algunas canas.
So you'll just need to inject your blood sample into this port on the cartridge, load the cartridge into the system, and the system will take care of the rest. Within 15 minutes, you'll get a pure population of the circulating tumor cells that's comparable to the tissue biopsy samples.
So we even have specialty vials, ones that have internal coatings. So for example, this one, if you have a liquid, it doesn't stick to the glass.
Hello and greetings from Boston, where I am at the Bio International Convention this week. This place is ground zero for life science deal making and learning. And I'm here on the convention floor, where there are over 20,000 registered participants, 46% of them international. I've been spending most of my time this week upstairs, where they have some smaller rooms where they're doing really interesting deep dives into making better clinical trials or AI for drug discovery. And while we're here, I thought we could take a look around and see what folks are up to. So one of the things I'm intrigued by is the booths that are named after countries and states. So let's get started there. Magdalena,
Yeah.
tell us about yourself.
So I'm representing the BioForum, the Association of Biotechnology Companies from Poland, and together with Polish Investment and Trade Agency, we are showcasing Polish biotech companies at the most important event for biotechnology. So under one roof, 20,000 representatives of biotech, and we're here. So
Beautiful. All right, I'm here with Rafaela with UK's Department of Business and Trade. Rafaela, why is the UK just a great place to do life science research?
Thank you. The UK has a very long history in life sciences and healthcare innovation. I think this is well recognized from the first antibiotics right through to the most recent innovation in mRNA vaccines, for instance, during the pandemic. The UK is generally recognized for its excellent science base, as well as spinning out companies. 1 in 3 spin-outs in life sciences and AI is in the UK compared to Europe. So a really strong base of companies. There's about 6,000 companies in the life science sector employing 300,000 persons. It is one of the priority sectors for the UK government, for the new UK government, and very much a sector that has a lot of commitment and support from this government.
All right, I am here with Rachel. Rachel, you are with Choose New Jersey. Can you tell us more about Choose New Jersey?
Sure. So thank you for having us today. My name is Rachel Cohen. I'm the Senior Business Development Officer for Choose New Jersey. Choose New Jersey is the leading nonprofit economic development organization in the state of New Jersey. We're really focused in on getting companies to choose New Jersey.
Awesome. So I was raised in New Jersey. I'm a Jersey native. I'm Jersey proud. Can you tell us why New Jersey is like the best place to do great biotech work?
Well, you're from it, so that's number one. But definitely, there's a lot of other reasons. But I would definitely say that we have a great location, central part of the northeast corridor between Boston and DC. So getting anywhere within our location is quite simple and easy. And because of that, we have great infrastructure. So we move people, move product very easily. So once you're landed in the U.S., landing in New Jersey, you can get to the third of the U.S. population within a day's drive. So really very, very convenient to many organizations. Second point would really be the workforce that we have in the state of New Jersey already. Think about it, we have big companies, we have great universities, you put them all together, great talent in the state. And then the last really kind of big point there is the quality of life in the state of New Jersey. So great, number one, public schools. So sending your kids to public school is fantastic. Great quality of life in the sense of the diversity in the state as well. So if you're an international company, you find people that look like you, eat like you, and dress like you in the state, which is a really fun thing to do. And then on the weekends when you're not thinking about your biotechs and things like that, hopefully we get a little time to get outside, from mountains to beaches to the river to pine barrens to farmlands to downtown cities, and being in the middle of the New York and Philadelphia corridor. You can't ask for more. So I really think New Jersey has it all.
I am sold. I'm coming back home.
I love it.
All right, thank you, Rachel.
Thank you. Thanks for having us.
All right, so Poland, the UK, New Jersey representing. What about all the cool equipment vendors? What are they up to? I'm here with Ville from MEPSGEN, and he's got a really great, interesting platform for organs on a chip. Can you tell us more about it, Ville?
Absolutely. So we have a automated system or platform to run microphysiological systems, organs on chips. We have different kinds of chips that we sell. So these are hard plastic chips. They have a compartment for running different kinds of biology. This is meant for organoid culture, which you can embed in a 3D ECM matrix here. And then we have channels for mimicking the vascular compartment here on the top. These are all run on the ProMEPS system that we have here. This is basically meant for automating your chip preparation workflows. So ECM coding, cell seeding, everything's automated. We have a liquid handler here for that. It also runs your chips. So this is our perfusion module that provides the quote unquote like blood flow or mimicking the vascular flow here. And then we have the TEER module, the transendothelial electrical resistant module that you can use to interrogate your chips.
Very cool. And your company is based where?
The company is headquartered in Seoul, South Korea.
Great. And if folks want to look you up, how do they find you?
MEPSGEN.com. Yeah.
Okay, beautiful. Thank you so much, Ville.
Thank you.
I'm here with Aaron at ReciBioPharm. This really interesting product caught my eye. Aaron, tell us more about what we're standing next to.
Yeah, so what it's actually standing next to is actually a new product that we have generated that has actually come out of a large grant from the FDA. So what we've actually decided to do is build a PAT skid. So we're essentially taking QC and putting it on wheels. And so it's actually, this device has actually 6 different PAT technologies, and it's actually all controlled by actually one software program that can actually monitor your process. And right now, so we've actually done that. So it's actually for RNA as the first test case. And right now we're exploring, it's actually doing this for MABs, it's actually ADCs, it's actually small molecules and et cetera. But what this allows you to do is basically it's actually centralize your analytics and put it on the production floor. And so we can also, it's actually modify this by swapping out decks, to put in actually additional technologies. So if you need, so it's actually NMR, for example, or flow cytometer, so that's something else that we can add to the skid so that we can cover as many of the CQAs as possible so that we can fully actually integrate manufacturing to reduce the cost and time to produce these therapies. It's actually so far what we've actually been able to do for RNA is actually take a 21-day process and actually condense it to a day to be able to go from IVT to fill finish.
Wow. Very cool modular design, great impacts on cost and quality. Thank you so much.
Thank you.
Appreciate it. All right, I'm with Eric at BloodScan Biotech, and he was just telling me about how when you have cancer and you need a biopsy, there's a liquid biopsy option, and this tool helps you do that in a really effective way. Eric, can you tell us more?
Sure. So this is a system where we use to isolate the circulating tumor cells from blood. So whenever a clinician wants to do a biopsy, but he couldn't access to the actual biopsy, then we can use this system instead. So normally when a patient goes through their journey of the cancer treatment, most of the time there won't be a second chance of getting biopsies, but still we want to know how's the cancer progressing during the treatment, and clinicians will want to analyze the cells. So the challenge here is that these circulating tumor cells, or CTCs, they are really, really rare in blood, about 1 in a billion of normal blood cells. And that's what we are good at. We isolate the CTCs from the blood using this called Labyrinth 1. And it's originally from our invention at the University of Michigan as a microfluidic chip. So we took the inspiration from the Greek mythology Labyrinth, where the monster Minotaur was isolated from the world. And when we are miniaturizing the maze into this kind of small structure, there are some fluid dynamics take place, most notably the lift force and ding force, which will focus the cells into a single streamline and then separate them by their physical properties. And we took that inspiration, so now we can isolate the cells from all the cancer stages from stage 0 to stage 4, from all types of solid tumors. We have over 15 publications covering from breast, lung, pancreatic, prostate, also all sorts of solid tumors. Any kind of analysis you can do on the tissue biopsy, we can now do on the circulating tumor cells. So it can be used in the early cancer detection, in precision medicine, and in cancer monitoring. And we even work with pharmaceutical companies to verify whether their drug will work on the patients.
Okay, beautiful. Eric, could you give us a quick run through of how the system would work in practice?
Sure. So this is the cartridge system and the machine. So in reality, you'll just need to inject your blood sample into this port on the cartridge, load the cartridge into the system, and the system will take care of the rest. Within 15 minutes, you'll get a pure population of the circulating tumor cells that's comparable to the tissue biopsy samples.
Amazing. Well, from Greek mythology to science reality, really cool technology. Thank you, Eric, for sharing that.
Thank you. My pleasure.
Well, that's a small glimpse into the people and the places and the hardware and technologies that I encountered over my week here at the Bio International Convention. As I reflect on the week that I've spent here and the one-on-one conversations that I've had and the panels that I attended, there are a few themes that come to mind. One is moving beyond AI's application towards just drug discovery and into the rest of the drug development process. For example, better managing the paper-based batch records that are often used in manufacturing, or developing digital twins in order to have better, faster, easier lab automation. The second is, there was ample talk about some of the structural challenges to innovating in this space, from the regulatory environment to the high cost of capital, especially now with higher interest rates and some of the capital-intensive outlays that are required in order to make something happen. At the same time, there were some really interesting, I think, innovative workarounds and ideas. For example, one person mentioned that one way of helping to drive down the cost of cell and gene therapies might be to actually have hospital systems develop those therapies in-house and actually develop them in a way where they can give them to the patients and pass the cost savings on to them. Finally, given the large amount of investment that we saw in biotech infrastructure in the COVID era and the slowdown that we've experienced since, now seems like a really good time in order to get a deal from a CDMO or contract manufacturer on developing a small batch of therapeutics. I wonder if there is a parallel here to the late 90s, early 2000s with the dot-com boom, where we saw companies like WorldCom that were laying down an incredible amount of broadband and fiber optics across the United States, infrastructure which they didn't get to make money off of, as a lot of them went defunct, but that ended up creating a foundation, a cheap and good infrastructure that a new generation of tech companies were able to take advantage of, tech companies that we know and use today. Could what was overbuilt in that previous cycle end up fueling this next one? Well, that's all for me here at the Bio International Convention. A big shout out to the team at Bio, who really laid out the red carpet for independent creators like myself, with providing access to the event and a media room that we could work out of. If you like this video and want to hear more from founders and innovators who are pushing the envelope on scientific discovery, check out our newsletter at discoveryengines.co or the videos on our YouTube channel, both of which are linked below. With that, thanks so much, and I'll catch you next time.